Hello, everyone. And hopefully you all are doing okay this evening. Today we are talking about glycolysis and gluconeogenesis and the role of aldolase gene expression. So thank you for joining us. And I am Miss Kapczynski. All right, let's go ahead and get started. All right, so a quick overview before we jump in and talk about some of these concepts is we are going to do an overview of glycolysis. So glycolysis is the process of a severe reaction that takes place. Let me rewind, a little tongue tap here. Glycolysis is the process of several reactions that take place that metabolize one molecule of glucose into two molecules of pyruvate, along with two molecules of ATP. So you can kind of see this in this picture here. So one glucose turned into two pyruvate with the addition of two ATP, which also benefits from that. So this is an anaerobic process because it doesn't require O2. So for example, Olympics sprinter in the picture there, that's Michael Johnson, and he requires the production of glycolysis to happen inside the body so that there is a source of energy that can be rapidly used. This is the anaerobic metabolism of glucose, and it's allows that very short, fast, intense amounts of energy to be created and released into the body for use. So in this case, Michael Johnson needs that when he's sprinting to get more energy to those muscles so that he can finish that really fast sprint. There are also other ways that pyruvate can be created, and this is anaerobically, which is through fermentation. So once it is fermented, it turns into lactase or ethanol. When pyruvate undergoes anaerobic condition, so that oxidation occurs to create CO2. In this process, it creates more ATP. So that's one benefit to that. So the history of glycolysis came from the Buchner family in 1897 when they were looking to extract yeast from free cells for therapeutic uses. So these extractions that they took when manufacturing the free cell extracts, they had to be preserved. So therefore, they didn't use any antiseptics. Instead, they tried sucrose, which is a common sugar that you're going to find in your kitchen as a preservative. And they found that sucrase became fermented into alcohol by yeast juice. So this discovery paired with previous scientist discoveries that were shot down, and it helped open the doors to modern biochemistry. So glycolysis also got its name glyc, which means sweet. So from the discovery of with that sugar, and lysis meaning dissolution. So if you look at the lactic pathway was fully elucidated, so meaning it was fully figured out by the 1940s, with many different scientists contributing to it. So a few of those scientists were Gustav Embeden and Otto Meyerhoff. So those two is scientists specifically, that is who that glycolysis pathway is named after. So you can see in the picture, I have a picture of both of the Buchner family members that contributed to that discovery in 1897 there. So kind of interesting little piece of history. So glucose can also be made from pyruvate, and so that lactic acid. This is a non-carbohydrate precursor process that is also known as gluconeogenesis. gluconeogenesis. So glycolysis and gluconeogenesis may seem like they are opposites, but they are two completely different pathways. However, they do have a few enzymes that they share and commonalities with each other. So gluconeogenesis occurs when there is an extra energy available and it is inhibited if energy is requires it, it can be stimulated by glycogen and is inhibited by insulin. The table here shows the steps of gluconeogenesis, and these steps are not reversible, but with specific enzymes, some of these steps can be skipped. So you'll see that some of these steps are, opposite reactions, but they are carried out by enzymes, specifically beta-directional enzymes. So we have if we're looking at this chart, I love charts for learning. So I really encourage you guys to include these in your notes because they are a great learning tool. But we will do some additional information with these in some later lessons and piecing those informational puzzle pieces together. So if you're looking at the enzymes, you have your pyruvate carboxylase. Again, all of our enzymes, they end in ase. So that's our first step. So the reaction is shown. It converts pyruvate oxalate. It requires ATP and our vitamin B7. And this is happening in the mitochondria, and it's stimulated by acetyl-CoA and inhibited by ADP. So that's where that first step in that pathway is coming from. And then our second step here, it's going to convert that, and it's going to require some G2P. And then cytosol and mitochondria is where it's occurring. Again, it's stimulated by glycogen and cortisol, and then it's going to be inhibited by that insulin. Our third step is fructose 1 and the 6 bisophosphate ACE. And this is can do that conversion of that protein. So the enzyme matches with that protein. And it's going to rate limiting enzymes in gluconeogenesis. It's happening in the cytosol citrate, and ATP is what it's being stimulated by, as well as fructose 1,6 bisophosphate. So, and then it's going to be inhibited by fructose 2,6 bisophosphate and AMP. So then in our last stage, we have our glucose 6 phosphate phosphase. And it's going to convert to G6P to glucose. Again, our end goal is to get to that glucose sugar. It's happening in the endoplasmic reticulum. So it's going to get that glucose to the blood. And it is stimulated by G6P and again, inhibited by insulin. So you can kind of see some of those commonalities there. So here you can see some of the side-by-side comparisons on how glycolysis and gluconeogenesis are different, as well as how they are the same. So if you're looking at location-wise, they are both happening in cytosol. Their gluconeogenesis does happen in the mitochondria, though. Again, oops, my bad. Rate limiting enzyme, they're completely different. So that's one of the differences that we're seeing. And then as well as stimulation for glycolysis, it's happening with the stimulation is insulin, and gluconeogenesis, it's going to be the glycagon. And then flipped when we're looking at the inhibition. So it's going to be completely flipped. So now it's going to be the glycogen that for glycolysis, and then insulin for the gluconeogenesis. When it is occurring, we have stimulated postprandial states, and then fasting states is when it's going to happen. So again, really great table, something that I encourage you guys to put in your notes. And then you have that as a resource on where those things are happening, and a side-by-side comparison on what those big differences are. Okay, involvement of aldolase. So the way that aldolase is involved in glycolysis and gluconeogenesis is when it converts 6 carbon products into 3 carbon products. So it takes fructose 1 and 6 bisophosphate to create G3P. So that's that glyceraldehyde 3 phosphate, and DHAP, which is dihydroxide acetone phosphate. And the creation of DHAP and G3P creates a response of generating NADH by systolic metabolism. And this then gets a reoxidation to NAD plus, causing a reduction in both DHA and P2G3P. And this includes glycolysis, and the reduction equivalent can still be used to create more generations across the protein gradient at the mitochondrial intermembrane. So it can do this by coupling and oxidizing G3P. The NAD plus regeneration is more influential during high glycolic flux. So G3P also causes another starting source for synthesis, causing glycolipids, which means that the G3P will need a constant supply of glycolysis for this to occur. So through this reaction, it increases ATP production, and the citric acid cycle increases. And this is because of the PP8 that is formed during the glucose carbons that come into the mitochondria. So the glycolipid fatty acid cycle has been researched and has connections with some of the human body processes, such as insulin resistance and synergy with post-Randall glucose. So those are some of those things that can be impacted in the body. There's different types of isoforms of aldolase, and the 3 different family members of aldolase are aldo A, aldo B, and aldo C. Each of the aldolase family members have a different gene that encodes them, and these 3 enzymes can be seen in human organs. So to break these down, I also have a picture to the right here to show that the aldolase, as well as aldo B and aldo C, which we'll talk about. So how these differentiate from each other. So first off, where can we see these being expressed in the body? So we know that they happen in different areas of the body. So aldolase A is in chromosome 16q22-q24. So where can that be seen? Expression-wise, where is this gene expressed? So it can be seen in embryos as well as adult muscle tissue. It has the lowest Km for F1 and 6BP catalysts when comparing to both aldolase B and C. So therefore, aldolase A has a lesser role in glycolysis, as well as the maintenance of glucose homeostasis. And there's a picture of some embryos here you can see. Aldolase B is expressed in the liver and kidneys. It can also be seen in the small intestines as well. and this is why it's commonly known as the liver type aldolase, or it can also be known as the fructose bisophosphate aldolase B. So when the scene, when this is dysfunctional or has a deficiency, it can lead to a heredity fructose intolerance. And this is also known as HFI, which is a rare disorder, but it can be seen. Therefore, it does not, what happens when this occurs, the body is unable to catalyze fructose, which then you'll get very sick. So vomiting, you'll have some problems eating as well. Worst case scenario, you can also get organ failure. And again, that's going to be the organs that it's expressed in. So it's going to be liver and the kidneys, and possibly death, because again, if your organs shut down, it's a very serious problem. And that's, again, if there's no treatment or prevention, if you know you have this rare disorder. And this is because that fructose 1-phosphate collects in the urine, and it's going to accumulate, and the affinity binding with phosphate acid. So it creates that affinity binding. This blocks energy metabolism in the mitochondria. All right, Aldolase C. So this is going to be expressed in the central nervous system. So more specifically, it's going to be seen in the cerebellum of the Purkinje cells, as well as the hippocampus and the Schwann cells. There are studies that have been identified that the Aldolase family has translocational situations that signal transduction during tumor progression, as well as other diseases. So kind of some interesting thoughts to think about as the Aldolase C is seen in the brain. So there can be some type of brain disorders. So that'd be kind of interesting to take a peek into later this week. So more on Aldolase B expression. Aldolase B expression can be regulated by transcription factors HNF1, HNF3, C/EPB, and DBP by having the protein binding sites added to the Aldolase B promote. Element D is part of the liver enriched proteins. This includes both C/EBP and DBP. So our CEBP forms heterodimers, and the CEBP and DBP are trans activators for the albumin gene. And then the DBP is a transactivator for the Aldolase B promoter and hepatotama. So that's the hepta G2 cells, and the CEBP is active in ladder cells. Both are active on the albumin promoter in hep G2 cells. On the other hand side, so the first transcriptor factor was our HNF1, and that's the first one that was studied and looked at. And so our gene activity is found, so it can be expressed in the liver and the small intestines, have been identified with the HNF1, and it's so that's that expression factor. So our HNF is also known as that POU protein, which is produced in the liver, small intestines, pancreas, and yolk sac, and of course the kidney. The HNF3 has different genes that can be looked at, and that they have 3 different. So they have an alpha, a beta, and a gamma. They all have similar binding and transcription activities, and they are transcription factors of the alpha-1 trypsin and the transthyretin protein promoters. The HNF3 isoforms are produced in the same area as the HNF1, except the kidney. So that's the big difference there. So they're different except the kidneys. HNF1 and HNF3 combined on an overlapping site. So this is between the NT, so the 126 and the 104. HNF3 counteracts HNF1, the dependent trans activation, and HNF3 does the same to DBP. So that's a little bit more on the Aldolase B expression there. And in the picture, you can see the overlap of the HNF1 and the HNF3. You can see that there's that overlap there. So this is where you're looking on chromosome wise. So you have your DE, your DBP, your C/EVP. So all of those different Aldolase B expressions. And here are my references that I used today. So we have some information and some really great research studies that looked at the different types of Aldolase, as well as the discovery and description of that process for looking at glycolysis and gluconeogenesis. So if you're curious, please take a peek at those. If you have any questions, please feel free to reach out. And I look forward to moving forward and discussing more on the topic of glycolysis and gluconeogenesis. Thank you for tuning in.